A carpenter ant leaves the trail 2 hours before she should. She does not stop when the pheromone road thins beneath her. She does not turn when two sisters tap her with their antennae, the standard colony handshake, the chemical ID check every worker answers 1,000 times a day. She walks past them. She climbs. The leaf litter drops away below her. Soil gives way to bark. Bark gives way to open stem. She moves upward with a slow, mechanical steadiness that looks nothing like foraging, nothing like scouting, nothing like panic. A healthy ant that loses the trail circles. Searches. Returns. This one does not circle. Something else is moving her legs. To understand what is wrong with this worker, you have to understand how an ant knows anything at all. She does not see the colony the way you see a building. She does not recognize her queen by face, her sisters by voice, her nest by shape. She recognizes them by scent. Every ant in a colony wears a thin, waxy coat of cuticular hydrocarbons on her exoskeleton, a chemical signature as precise as a fingerprint, but shared across the entire nest. When two workers meet, they touch antennae. If the hydrocarbon profile matches, the stranger is a sister. If it does not, she is an intruder, and the response is immediate coordinated violence. This is not a preference. This is the architecture of reality. An ant's sense of smell governs who she feeds, who she fights, who she dies for, and whether she recognizes herself as healthy or sick. Strip that sense away, and she does not lose a convenience. She loses her entire world. The colony is not held together by loyalty. It is held together by chemistry. And chemistry can be rewritten. The obvious explanation for the climbing worker is disease. A sick ant. Confused. Stumbling off the trail. But sickness in ants does not look like this. A sick worker slows down. She stops foraging. If the infection is severe, her colony detects the change in her cuticular hydrocarbons, her scent shifts, and her sisters respond. In a functioning colony, social immunity kicks in. Healthy workers groom the infected ant aggressively, or isolate her, or carry her body out of the nest before the pathogen spreads. The system works. It has worked for over 100 million years. So why is this worker climbing with such precision? Why is she not slowing down, not collapsing, not being dragged away by her sisters? And why did none of them raise the alarm? The worker is carrying a passenger. Days earlier, somewhere between 4 and 10 days, depending on the species, a single Ophiocordyceps spore landed on her exoskeleton. Using specialized enzymes and raw physical pressure, the spore drilled through her armor and entered her body cavity. Since then, fungal cells have been multiplying inside her, feeding on non-vital fluids and soft tissue. Here is where the old story gets it wrong. For decades, the popular version said the fungus invades the ant's brain, eats it, takes over the controls. That is not what happens. Three-dimensional scanning electron microscopy has shown that Ophiocordyceps infiltrates nearly every part of the host's body except the central nervous system. The brain is not consumed. It is surrounded. Fungal hyphae weave through the ant's muscles, particularly the mandibular fibers that control her jaws. The fungus secretes targeted compounds, ergot alkaloids, neurotoxins that act directly on muscle tissue, severing motor control from the brain's commands. The old myth said the brain was destroyed. The research says the brain is structurally intact, walled off by fungal tissue, while the body answers to something else entirely. That distinction is not a relief. It is the mechanism that makes the final stage possible. Under chemical control, the ant does not wander randomly. She exhibits phototaxis, a compulsive movement toward light, upward, away from the dark safety of the forest floor. Her coordination degrades. Researchers describe a stagger syndrome in the late stage, motor function fragmenting, the ant lurching upward on legs that no longer fully belong to her. Her chemical world has been erased. The olfactory dampening strips away her ability to smell her own colony, her own infection, her own decay. This is the part that defeats the colony's defenses. A healthy ant that detects lethal infection in herself is biologically programmed toward altruistic exile. She leaves the nest, removes herself from the population, protects her sisters. But the fungus has blinded her to her own condition. She cannot smell what she has become. So she stays passive. She does not self-exile. She climbs. When she reaches the right height, a specific microclimate of temperature and humidity ideal for fungal reproduction, the fungus forces the final act. Her mandibles lock onto the underside of a leaf vein. The jaw muscles hypercontract. The grip is permanent. She dies there, alone, clamped to a leaf she had no reason to climb. Over the following weeks, a fruiting body erupts from the back of her head. It releases spores. The spores fall onto the trails below. The cycle starts over. The colony is not defenseless. Ant societies have been fighting fungal parasites for tens of millions of years, and the immune response is not individual, it is social. Healthy workers groom each other constantly, scraping spores from exoskeletons before they can drill through. If a body is found inside the nest, dedicated undertaker ants carry it out and deposit it far from the foraging trails. Some species avoid entire zones of the forest floor where spore density is high. The colony treats infection as a structural threat, not a personal one. But Ophiocordyceps has evolved around these defenses. The olfactory dampening, the chemical blinding of the host's sense of smell, means the infected worker does not trigger the colony's detection system in time. She smells normal long enough for the fungus to finish its work. By the time her body is clearly wrong, she is already climbing. The colony can fight what it can detect. The fungus makes sure it is not detected until the weapon is already aimed. That is one version of zombification. The body hijacked, the brain structurally spared, the colony's immune system outmaneuvered by a parasite that erases the ant's ability to know she is infected. But there is another version. And it is not a fungus, it is other ants. Ophiocordyceps takes one ant. It uses her body, kills her, and moves on. What some ant species do is worse in a different way. They take entire generations, and they do not kill them. They reprogram them. The formal term is dulosis. A slave-making ant species raids the colony of a different species, fights through the defenders, and steals the brood, the larvae and pupae. The stolen young are carried back to the raider's nest. When those pupae emerge from their casings, the first 10 days of adult life, they enter a critical neurological window. During that window, they learn the chemical identity of whatever colony surrounds them. Because they are surrounded by the slave makers, they imprint on the wrong signature. The result is permanent. The stolen ants do not know they have been stolen. They groom, feed, forage, and defend the colony that abducted them. If they are ever reintroduced to their biological sisters, they attack them because the chemical profile does not match. Their real family smells like the enemy. Their captors smell like home. This is not metaphor. This is measured, documented chemical reprogramming. The prison has no walls. The chains are hydrocarbons on a waxy shell. The species that practice dulosis have evolved tools as specialized as any predator's claws. Formica sanguinea, the blood-red slave maker, is a facultative parasite. It can survive alone, but it chooses not to. When it raids a colony of Formica rufa or Formica cinerea, the attacking workers deploy propaganda pheromones, volatile compounds, decyl acetate, dodecyl acetate, tetradecyl acetate sprayed directly onto the defenders. In the Formica sanguinea group, one species, Formica subintegra, carries approximately 700 micrograms of these compounds in a single worker. That is 10% of her entire body weight, synthesized internally and stored in a massively enlarged Dufour's gland. For the attackers, the compounds act as a coordination signal, a chemical war horn that synchronizes the assault. For the defenders, the same compounds register as an overwhelming, supernormal alarm signal. The result is instant mass panic. The defenders scatter, abandon their defensive positions, leave the brood exposed. The raiders walk in and take the children. And here is the detail that twists deepest. The enslaved ants do not just serve. Observations of mixed Formica sanguinea colonies show that enslaved workers, ants stolen as pupae, raised in the wrong nest, join subsequent raids. They march alongside the column that kidnapped them. They pick up dropped pupae from the target nest and carry them back. They help enslave their own species. In the nests of the slave-making ant Protomognathus americanus, the enslaved workers are Temnothorax longispinosus, small, quiet ants tasked with foraging, cleaning, and raising the parasite's brood. They cannot escape. They cannot reproduce. They cannot return to colonies that would now attack them on smell alone. By every visible measure, they are fully integrated, fully compliant tools of the slave-maker queen. But inside the nursery, something else is happening. The enslaved nursemaids do not rear the parasite's young with full commitment. They neglect some pupae. They dismantle others. Research conducted across colonies in New York, Ohio, and West Virginia found that enslaved workers systematically destroy the brood of their captors. On average, only 45% of the slave-maker's offspring survive to adulthood. The rest are killed, quietly, steadily, by the very workers assigned to protect them. The enslaved ants gain nothing from this personally. They are trapped. They will never leave. But by reducing the slave-maker population, they cripple the colony's ability to raid the following season. Fewer raiders means fewer stolen pupae. Fewer stolen pupae means the free-living colonies nearby The colonies that contain the slave's true biological sisters survive. It is not a conscious uprising. It is a genetic reflex shaped by millions of years of pressure, expressed as targeted destruction from inside the enemy's nursery. The slave makers stole the workers. The workers are destroying the slave makers' future. And neither side chose this war. 34 million years ago, in what is now the Baltic coast, an ant with specialized cranial morphology was sealed in amber. Researchers extracted her with X-ray microtomography and found physical hallmarks consistent with social parasitism. The arms race between host and parasite was already old when mammals were still small. It is still running. In a temperate forest in North America, a carpenter ant clamps her jaws to a leaf and dies with a fungus erupting from her skull. In a meadow in Europe, a stolen pupa opens its eyes in the wrong nest and spends its life defending the species that took it. In a tiny acorn in the northeastern United States, a raider 2.5 millimeters long walks through an enemy colony without triggering a single alarm, because her scent says she belongs. The fungus does not think. The slave maker does not plan. The rebellion is not a decision. But the chemistry is precise. The chemistry is always precise. An ant knows her queen, her sisters, her enemy, her home, her duty, and her death, all by smell. Every bond she has is molecular. Every loyalty is a compound on her skin. The horror is not that these systems break. The horror is how little it takes to rewrite them. One spore, one stolen pupa, one scent that says you belong here now. The body follows. It always follows.